Hello everyone, this is the Theoretical Doctor and welcome back to my channel. If you're new here, welcome! This is a series on the chapter Transport in Animals and Plants of Chapter 8 of the STPM Semester 2 Biology series. Check out the full playlist on my channel. If you want access to flashcards on this topic and other videos, do click the link below. It is available on my website as well as slides on this topic. In this video, we will be continuing on translocation in phloem, phloem loading and unloading, as well as pressure flow or mass flow hypothesis. The subtopics we will be touching on would be the structure of phloem, translocation in phloem, as well as the phloem loading and unloading, and pressure flow or mass flow hypothesis. Structure of phloem. So the phloem consists of sieve tubes and companion cell. This part over here are the companion cells, and this part over here are the sieve tubes. So sieve tubes are long cylindrical structure consisting of narrow living elongated sieve elements joined together. So the sieve tubes are actually living cells, and the companion cells are smaller and shorter than sieve tube elements. They supply ATP required for active transport of sucrose and amino acids into the sieve tubes. So the phloem sieve tubes are arranged end to end like this to form a continuous cylindrical stack called sieve tube. Their end walls are called sieve plates. This part here, these are the sieve plates, which are perforated, so there are holes in them, but not completely broken down to separate them from the adjacent cells. So these perforations allow cytoplasmic strands to pass through to connect one cell with an adjacent cell. So these are the cytoplasmic strands. The perforation is called plasmodesmata. The companion cells form intimate contact with phloem sieve tube cells through the plasmodesmata. So they're very closely linked together like this, if you can see over here. Every sieve tube needs a companion cell to stay alive. They have nucleus as well as other organelles, and they are living cells. So the companion cells, they do not transport organic substances, but they control the activity of the unnucleated phloem sieve tube cells. So these sieve tube cells, actually, they don't have nucleus, they don't have anything. Literally, their function is just to transport in the plants. So they need this companion cell. They're always together. So that's how you remember companion cell. You need company. So they're beside each other always. They come in pairs. They have to be together always. Translocation in phloem, phloem loading and unloading. Translocation is the transport of organic substances such as the soluble products of photosynthesis, amino acids, hormones, and other metabolites in the vascular plant from the leaves, which is the source, to the sieve tubes to be carried out to other parts of the plant, which is the sink. So movement here is from source to sink. Translocation is carried out in the vascular tissue known as the phloem. It is an active process, and the energy required comes from companion cells, as you can see in the previous slide. So source. Source is the region in the plant where photosynthesis occurs and organic solutes are synthesized. Example of source is green leaves of plants. Over here, organic solutes such as sucrose and amino acids are then loaded into the sieve tubes of the phloem. Now sink. Sink is an area where organic solutes are unloaded from the sieve tube where it comes out. Source is where it goes in. Sink is where it comes out. Examples of sink would be the growing shoot and root regions, developing flowers, fruits, and storage organs such as the tubers and bulbs. Sugars are synthesized during photosynthesis in the mesophyll cells of leaves. A chemiosmotic mechanism is involved in the active transport of sucrose. Sucrose is co-transported with protons into transverse cells and sieve tubes. So there are four hypotheses explaining the translocation in phloem. First is the pressure flow or the mass flow hypothesis, electroosmosis, cytoplasmic streaming, and peristaltic waves. However, in this video, I would be focusing solely on the pressure flow or the mass flow hypothesis. The pressure flow or the mass flow hypothesis. It takes place in 3 stages and involves a combination of active transport and mass flow. So the first stage, it occurs at the leaves where photosynthesis is carried out by the mesophyll cells to produce the organic substances. So first glucose is produced, but after that it condenses into sucrose because it's more suitable to be transported in the plant. So the source would be the leaves. That is where phloem loading occurs. Organic solutes are actively loaded into the sieve tubes, and it's carried out by the modified companion cells, and the modified companion cells have many mitochondria and internal projections of the cell wall, which then increases the surface area for transport. So these modified companion cells are called transfer cells. They use active transport mechanism to load sucrose into sieve tubes by co-transport. The high concentration of sugar in the sieve tubes causes a lower, more negative water potential in it. Consequently, water enters the sieve tubes by osmosis from the neighboring xylem, and a high hydrostatic pressure is created inside the sieve tubes. So remember, water moves from an area of high water potential to an area of low water potential. So increasing the concentration of sugar initially into the leaves will then cause a low water potential, and consequently water then moves in through osmosis. So that's the first stage. Now the second stage, organic substances are translocated in the stem from the source to the sink by mass flow. So sink are like stem tubes, taproots, bulbs, fruits, and seeds. It is a region in a plant where organic substances are translocated from the source and they are used or stored over there. So the concentration of sucrose in the sink is lower than in the source or the sieve tubes because sugar is continuously being consumed and converted into starch to be stored. This allows sucrose to diffuse out of the sieve tubes down the concentration gradient, and it ensures a continuous flow from the source to the sink. So remember, first in the source, sugar is actively transported, right? So it will create a high concentration of sugar at that region. Now, this area in the sink, the concentration of sucrose is low because they are actively being removed. They are actively being unloaded. So therefore, the sucrose is able to move passively from the source to the sink, because in order for it to move passively, the molecules or the substance has to move from an area of high concentration to an area of low concentration. So therefore, sucrose is continuously being moved throughout the plant. This is the Munch model by Ernst Munch, 1930. So this actually explains the pressure flow mass flow hypothesis. So there are two osmometers over here. Osmometer A represents the leaf cells or the source, and osmometer B represents the sink. So remember, the translocation occurs from the source to the sink. So the connecting tube between the two osmometers over here is the phloem sieve tubes, and the connecting tube over here between the two water containers represent the xylem vessels. So water flows passively from osmometer A to osmometer B, carrying the solute mass with it. So when it flows from here A to B, it is using the mass flow. In osmometer A, water flows into it from a region of high water potential to a region of low water potential by osmosis, and this will create a high hydrostatic pressure in A. This is because the low water potential is caused by the presence of concentrated sucrose solution. So remember, initially I mentioned that sugar or sucrose is actively pumped into the source. So that area will have high concentration of sucrose. So when there is a high concentration of sucrose, there is low water potential. So therefore, water over here, the water potential over here is very high, and then it will flow passively by osmosis. Water will then move from an area of high water potential to an area of low water potential. Remember, there's high concentration of sucrose over here. They can't go anywhere. When this water moves in, it will create a pressure, and the pressure is high. Remember here, water flows passively from osmometer A to osmometer B. Now this part over here, B, which represents the sink or the root cells, it contains dilute sucrose solution. Why? Because sucrose is continuously being unloaded from the phloem, right? Remember, in sink, sucrose is continuously converted into starch. So this part over here will have dilute sucrose solution, and water potential is higher, meaning there's more water over here. So because this area has high water potential and this area has low water potential, the water will then move from an area area of high water potential to an area of low water potential. So water is continuously moved out by osmosis. Because over here there is water plus the sucrose, there is high pressure, it will push, because remember that whole solution can't go anywhere, so it will push it over here and water will keep continuously moving out due to the pressure, which is due to the hydrostatic pressure gradient. So now when the water comes out, what happens? It's a cycle. They will be redistributed through the connecting tube, which is the xylem vessels between the two water containers. So in the plant, the high hydrostatic pressure in the sieve tubes is produced by an osmotic influx of water due to the accumulation of sucrose during active loading. This high hydrostatic pressure then pushes water and its mass from one sieve tube to another through the plasmodesmata. So this is just an easy representation of this pressure flow mass flow hypothesis. And we enter the third stage. So sucrose and other organic solutes are actively unloaded at the sink, involving living companion cells and energy, where sucrose may be used for metabolism or converted into a storage product. So when sucrose is being loaded into the phloem sieve tube, it uses ATP. When it is unloaded, when it is removed from the sieve tube at the sink, ATP is also involved. So to put in, you need ATP. To remove, you also need ATP, and the rest will just flow passively, which is the water. So water follows the organic solutes out of the sieve tubes, reducing the hydrostatic pressure and maintaining the pressure gradient from the source to the sink. The return of water from the roots or the sink to the leaves or the source through the xylem vessel is brought about by the upward transpiration pool. So remember, pressure flow mass flow hypotheses have 3 stages. But if it's difficult to remember, you just need to remember the Munch model. It simplifies all the stages for this pressure flow mass flow hypothesis. So come back here, pause the video, take your time to understand it. Because it is a hypothesis, there are evidences that support and evidences that are against this. So evidences that support the pressure flow hypothesis. First, the movement of organic substances in the phloem is too fast to be caused by diffusion. It involves energy and hydrostatic pressure gradient or concentration gradient, which has been detected between the leaves and the roots of various types of plant. Second, the concentration gradient between the leaves and roots disappear if the leaves are removed. So when the leaves are first removed, fluid flows fast from the cut leaf stalk. It shows that the phloem sieve tubes in the stem were in fact under great pressure. That's why water is able to come out from there. So evidences that are against the pressure flow hypothesis. First, it is difficult to make measurements of transport in the phloem due to disruption caused to the phloem. In the phloem flow hypothesis, substances cannot flow in opposite directions in the same sieve tubes. So the thing is that, remember in the Munch model, I mentioned that the whole mixture will go to osmometer B because, well, it can't go anywhere. So it shows that it moves in one direction, right? But in actual fact, the phloem contains many sieve tubes and different solutes could travel in opposite direction. It's a two-way highway. It's not just a one-way road. So the different solutes could travel in opposite directions at the same time in different sieve tubes with different sources and sinks. Third, the presence of sieve plates between the sieve tubes hinder mass flow. Fourth, the hydrostatic pressure measured in sieve tubes are insufficient to translocate organic substances. And lastly, translocation of solutes in the plant is influenced by temperature and metabolic poisons, which indicates that the translocation of organic substances is not a passive process. If you've reached this point, thank you very much. Hit the like button if you found it helpful, as it does help in reaching out to others as well. Please support my channel by pressing the subscribe button so I will know, and it gives me motivation to continue producing content like this in the future. And finally, share this video to those who might find this helpful. See you in the next one.